What's happening, folks? Arush here, and today we're talking about passive cell transport, or basically how do cells move things around without any energy. And if I take a look at these two pictures here, and I ask you to identify which picture shows an individual being passive, that would be this picture here with the cat. Okay, this cat is being very, very passive, and what that means is it's not exerting a lot of energy. And in contrast, this athlete is exerting lots and lots of energy, or ATP. So today, passive cell transport, again, is moving things around without energy or without ATP, as we've talked about. Okay, so let's go and get into this. First, some key vocabulary to understand passive transport. First, passive transport means without energy. Okay, passive transport without energy. Next, we have diffusion, and what diffusion is, it's the movement of molecules from an area of high concentration to an area of low concentration. Okay, so molecules are actually going to move basically from where there's a lot of it, and it's going to move to where there's a little bit of it. And I'll show you this in just one moment. Next, we have the word concentration gradient. What a concentration gradient is, it's when there's a difference in molecule concentration. And the exact opposite of this word, the exact opposite of concentration gradient, is equilibrium. And what equilibrium means, there's an equal amount or an equal concentration of molecules. What I want you to do is go ahead and put the video on pause right now and copy these words into your notes. Any slide that has this pencil, put it on pause. Whoops. Put it on pause and make sure this makes it into your notes. Okay? Okay, so let's get into what diffusion actually is. So, like I said, diffusion moves from high concentration to low concentration. So take this for example. Let's say I'm in the classroom and I take a can of Febreze and I spray that Febreze in one corner of the classroom. And there's lots and lots of Febreze molecules now. What's going to happen is those Febreze molecules are going to move from high concentration, basically where there's a lot that I sprayed, to where there's none that I sprayed. Okay, so those are going to start to diffuse. And if I go ahead and pause right now and I take a look at the classroom, I would say there's a concentration gradient. There's a different amount. There's a high amount of Febreze here, and there's no Febreze over here. So there's a concentration gradient. And what's going to happen? All of these molecules are going to diffuse from high to low concentration. Okay? And this is still a concentration gradient because there's still a lot of molecules here, and there's none over here. Okay, so there's still a gradient. They're going to diffuse until there's an equal amount, or until we reach equilibrium. Okay. So anywhere in the room that I move, I would still sense or smell the same amount of Febreze as anywhere else. Okay, so that's equilibrium, where there's an equal concentration. If I take a look at this picture, okay, this is an animation of Black Friday shoppers. I would see that there's a high concentration of shoppers here, and before the doors open, there's a low concentration of shoppers inside the store. Well, once the store opens, there's diffusion. I would say this is diffusion from high to low concentration. Okay? And why is this an example of passive transport? Okay, why am I using this as an analogy to show you the movement without energy? Well, take this for example. Let's say you're in the crowd, doors open, and you start running in. That's going to be pretty easy to do. Okay? But now if you start running in and you stop and say, oh geez, I forgot my wallet in the car, and now you have to run against the crowd, that's going to require lots and lots of energy. And that's actually called active transport, which we're going to talk about in the next lecture. Okay? So moving with diffusion, moving from high to low concentration is very, very easy to do. That's the point I'm trying to get at. So I want you to copy this into your notes as well. Label this diffusion. Show me that there's the movement of molecules from high to low concentration, from high to low. And I want you to think for a moment. Go ahead and pause and take a look at these three pictures. Which one of these pictures, or which of these pictures, shows a concentration gradient? It would be these two, because there's a different amount of molecules in the whole space. Okay, so that's a concentration gradient. Now, which one of these pictures shows equilibrium? Well, that would be this picture here, because everywhere there's an equal concentration or an equal amount of molecules. Okay? please make sure this makes it into your notes as well. This is just a quick little sketch. And now before we move on, we have to go over a few more terms. So we understand what diffusion is, but now we have to know what osmosis is. And osmosis is very, very important to biology, and it's actually a special type of diffusion. It's the diffusion of water, okay? It's the diffusion of water across a cell membrane. So this is how all of our cells get fluid. Basically, we've talked about, the cytoplasm in our cell is a mixture of proteins and water. Well, how do we get water into our cells when we drink it? All of that water is going to diffuse into our cells by osmosis, okay? A couple other terms that we have to know is what a hypertonic solution is. This prefix right here, hyper. Hyper means in excess, or a lot of, or higher amount, okay? So a hypertonic solution is when there's a higher concentration of solutes in a solution. And this will all make sense as we go through the video. Next we have hypotonic solution. That prefix, hypo, means low, okay? So there's a lower concentration of solutes in a solution. Okay, so hyper means there's a higher amount. Hypo means there's a lower amount. And now we have a third one, and that's isotonic solution. This prefix, iso, means there's an equal concentration, or there's the same concentration, okay? So let's take a look at how all of these are related. Osmosis, hypertonic solution, hypotonic solution, and an isotonic solution. Let's see how they're all related. Oh, I'm sorry. And the last thing that I want you to know, please put this in your notes, is salt sucks. And this will all make sense in just a moment. So first, osmosis, diffusion of water across a cell membrane. So as I mentioned in the past, water molecules are very, very tiny, and they're actually so small they could fit across the little crevices of the cell membrane. No problem at all, okay? And this is what diffusion is. This water is basically squeezing in between each phospholipid, okay? And that's how water is transported throughout all of your cells, is osmosis, okay? And now we have these two words, hypotonic and hypertonic. So I want you to picture this. All of these little blue dots, we'll say that's water. Whoops. We'll say those are water molecules. But now let's say I add salt or a solute, okay? A solute is anything that dissolves in water. Specifically, I'm going to talk about salt, okay? Just easier for this class. So let's say I add salt to one side of a cell membrane. Well, I want you to know this concept. Salt sucks. So what's going to happen is where there's a lot of salt, okay, we'll say this is the salt on one side of the cell membrane, that's actually going to pull the water molecules across, okay? So the water is going to be attracted to that salt, and it's actually, more water is going to move across the cell membrane, okay? So where there's a higher concentration, the water is going to be attracted to that area, okay? Now, we have salt on both sides, but now we have more salt on one side still. So this would be a hypertonic solution when I compare it to the right side. The right side is a hypotonic solution when I compare it to the left side. Now, where is the water going to go? Well, remember, salt sucks. So water's going to go to the left side. More water's going to go to the left side, okay? And now what if there's an equal amount of salt or an equal amount of solutes on both sides? Well, there's still going to be movement of water, but it's going to be equal, okay? And this is isotonic solution. What does iso mean? Iso means the same or equal, okay? So if there's an isotonic solution, the same amount of water that moves to one side equals the amount moving to the other side, okay? And let's see what is this actually, what effect does this have on a cell? So now let's say all of these purple dots, all these purple molecules, we'll say that is salt. And I want you to think for a moment, where is water going to go? Into the cell or out of the cell? And remember, this is a hypertonic solution. There's more solutes or there's more salt outside of the cell when I compare it to the inside. Well, since there's more salt outside, water's going to leave the cell, and that cell is actually going to shrivel up. Okay? Now, you may have heard if you consume too much salt or too much sodium, that will dehydrate you. And that is true. And this is the reason why. Because when you eat a lot of salt, that's actually going to pull the water out of your cells. Okay? And if you've ever, seen snails or seen anybody put salt on a snail, it actually kills the snail. And the reason why is you're putting a hypertonic solution on its slimy skin, and that's actually pulling the water out of its cells. And that's dehydrating the snail, and that's why the snail will die. Okay? So now let's see what happens to a cell in a hypotonic solution. Now think about this. Where is there more solute, or where is there more salt? Inside of the cell or outside of the cell? There's more salt inside. So where's water going to go? Water's going to go inside of the cell because salt sucks. So we have more water goes inside of the cell, and that cell's actually going to expand or swell up, okay? And if there's a big enough difference, this cell will actually lyse or split open or pop, okay? Because it expands too much. Okay, so this actually isn't very good for, in some situations. And now, if we look at an isotonic solution, where there's an equal amount or an equal concentration of salt inside as there is outside, well, that cell's not going to move at all. The same amount of water that leaves equals the same amount of water that enters, okay? So this is what we want. We want an isotonic solution around our cells. So take a look at these pictures. Picture A, is that a hypotonic solution, hypertonic, or isotonic solution? That would be hypo, because there's a low amount of solutes around the cell. Okay, there's a low amount. If I look at picture B, is that cell in a hypertonic, hypotonic, or isotonic solution? That would be a hypertonic because there's more solutes outside of the cell. Okay. And lastly, picture C, is that a hypo, hyper, or isotonic? Well, since there's the same or equal amount of solute concentration, that would be an isotonic solution. Now I want you to think. Go back to picture A. What's going to happen to that cell? Well, salt sucks. So more water is going to go into the cell. Wrong picture. More water is going to go into this cell, and this cell is actually going to expand. Okay, it's going to get bigger. Look at picture B. Again, salt sucks. So water is going to leave the cell, and this cell is actually going to shrivel up. Okay? And picture C, it's in an isotonic solution, so this cell stays equal. This cell will not lose or gain any water or any mass. Okay? So, take a look at this picture. Would this be a hyper, hypo, or isotonic solution? This would be hypotonic solution. And what's going to happen to this cell? Is it going to shrivel up, expand, or stay the same? Well, water's going to move into this cell, and it's actually going to expand. Okay? Take a look at this picture. Is this cell in a hypo, hyper, or isotonic solution? Well, since there's more solutes, or more salt outside, when I compare it to inside, it's a hypertonic solution. Now, what's going to happen to this cell? Is it going to expand, shrivel, or nothing? Water's going to leave this cell, and this cell is actually going to shrivel. Okay? And finally, this last picture. Identify. Is this a hypertonic, hypotonic, or isotonic solution? Since there's an equal amount, this is an isotonic solution. Now, what's going to happen to this cell? Expand, shrivel, or stay the same? Well, since there's the same amount of water moving in as there is moving out, this cell will stay the same size, okay? Now, again, let's go back to this question. Why is this important? Well, if you've ever known anybody to have an IV, if you look at that bag, this bag isn't actually distilled water, okay? It's not just regular tap water. This is very, very specific. It's 0.9% sodium chloride, or there's 0.9% salt in there. Now, why is that? Well, what the doctor wants to do is, your doctor wants to make sure that this concentration of sodium chloride, or salt, is the same as your blood. It's the same as your body. Because if they were to put a hypotonic solution into your veins, where there's no sodium at all of your red blood cells, they'll swell up. Okay? What if they were to put a hypertonic solution? Let's say they were to put like 5% sodium in that bag. What would happen to your blood cells? Well, your blood cells would actually shrivel up. All the water would be pulled out of your blood cells, and they would shrivel up to little raisins. Okay? So the doctors make sure that you get 0.9% sodium chloride, so that matches the same concentration as your blood, and your blood cells maintain their shape. Okay? And what actually can happen in a hypotonic solution, again, is that your cells could lyse, and they could pop because they expand so much. Okay? So it's very, very crucial to maintain an isotonic solution. Okay? So, real quick recap. We talked about diffusion, which is the movement from high concentration to low concentration. Again, if I look at these three pictures, these two pictures here show a concentration gradient because there's a difference in molecules. This last picture on the end shows equilibrium, because there's an equal amount of molecules in the whole space, okay? And one last thing, diffusion is passive. Does not require any energy, okay? Next, we talked about osmosis. It's diffusion across the cell membrane. It's diffusion of water across the cell membrane. We talked about a hypertonic solution, where there's more solutes or more salt outside of a cell. Hypotonic is when there's less solutes outside of a cell. And lastly, an isotonic solution is where there's an equal amount of solutes or salt outside of a cell. All right, that was it. I apologize for this being a little bit longer than normal, but I will see you guys next time. Talk to you later. Bye.